Welcome back, friends. We are talking about cell cycle control. They actually work. That in checkpoints are timestamps during cell cycle when a cell try to decide whether they will divide or not based on everything is okay inside the cell or not. That means if the cell size is okay, all the DNA is replicated or not, whether the chromatids are properly attached to the microtubules or not. So there are checkpoints in the cell cycle to check that. Now when they check it and find something is okay, everything is fine, then they will start to divide. But the proper cell division to occur, they require multiple proteins to work together, who will progress the cell to the next level or to the next phase. Now for that reason we rely on two proteins, cyclins and CDKs. Cyclin is a protein which is always produced inside the cell in small amount, but depends on the situation it will be activated or it may not be activated. But CDK is cyclin dependent kinase, which will actually do all the functionality because it is a kinase enzyme which can phosphorylate other proteins to make them active or deactive, right? So here CDK, why it is CDK? Cyclin dependent kinase. So the activity of this kinase protein depends on cyclin. See, if there is no cyclin, CDK will not function properly. Okay. So in every different stages of cell cycle progression, they requires this CDK as well as cyclin combinations. In each case, there is a CDK cyclin combinations which are helping the cell to progress to the next phase of the cell cycle. Now if I talk about the cell cycle in a whole, let me draw the image again. This is the cell cycle. And in this phase, let's draw, say, let's say this is a inner interphase and this is the mitotic phase, M phase, and this is the whole interphase. In the interphase, let me divide it into three different sections, say G1 phase, S phase and G2 phase. While in the M phase, we will divide it into four: prophase, metaphase, anaphase and telophase. Four different phases in the M phase also. This is the overall cell cycle for a mitotic cell division. Now if you look at here, it will move from this direction. If I put the arrows, the clockwise manner. Okay. So it will start from G1, go to GS, then G2, then like that. In the G1 phase, they will check whether the cell attains proper cytoplasmic content and ready to divide or not, whether the cell has all the enzymes for the replication of the DNA or not. In the S phase, they will replicate the DNA. In the G2 phase, they will check, at the end of G2 phase, they will check whether the replication worked properly or not, and also they will check for whether the proper synthesis of DNA is there or not. Some RNA synthesis is required in the G2 phase, which is already been done, and also they will check the size of the cell and maintain the size, and once they think everything is fine, then only they will progress to the M phase of the cell division, because M phase is the actual phase where the cell will divide, but before that the cell will prepare for the division. Now during different stages we have checkpoints. We have a checkpoint between G1 and S phase, we have a checkpoint between S, G2, I told you, but also you can put it the same way like G2M checkpoint, either between S, G2 or G2M. There are multiple researches going on, and actually these checkpoints we think of ourselves to understand more. So we know that there is a checkpoint probably is G2 and G2M, but ultimately the checkpoint has the same functionality to check whether the substrate whether the DNA replicated properly or not. If the DNA replicated properly and cell size is okay, then only they will pass through the M phase. So that is why it will check at G2M phase, and ultimately there is an M phase checkpoint. At the M phase checkpoint, they will determine whether the microtubules are properly attached to the kinetochore or the chromatids or not, whether the metaphase alignment of the chromosomes are perfect or not. Okay. So in all this case, if everything is fine, if those things are okay, if they get a positive signal, then only cell will divide. Okay. So for that reason, they require certain type of cyclins and CDK to perform that job of pushing it to the next level if the things are positive. Okay. Now let's say in this case, every time it passes from a G1A, say G2, all these cycles, they recruit certain type of combination of CDK and cyclins to drag it in the next level. Okay, but the beginning we have a G1S cyclin which acts at this particular junction and put the cell from G1 phase to the S phase. We have a SG2 cyclin which acts here, or S phase cyclin acts here, which will bring the cell to the next G2 phase. We have a G2M cyclin which works here, or M phase cyclin. So mainly three types of cyclin based on the three types of checkpoints which will help them to go to the next level. We have a G1S type of cyclin, so let me write G1S cyclin, and we call this cyclin as a D, cyclin D. Okay, cyclin D. And the cyclin that we work here in the S phase is cyclin E. Cyclin E, so let me write CDK complex. Here cyclin A CDK complex, at the end cyclin B CDK complex. So there are cyclin and CDK partners, right? CDK and their cyclin partners in each different stages are required to push the cell cycle to the next level, right? So in this case, this cyclin dependent kinases cannot function without cyclins. So here at the very beginning, G1S cyclin, you can also, if you don't remember the name of where is D, E, A and all these names, you can just tell it as G1S cyclin, S phase cyclin, G2M phase cyclin and an M phase cyclin. These are the names you can also call it, and actually I want to call it that name because I don't remember all the tiny details and stuff. So the thing is, here at the very beginning at the G1S phase cyclin, those partners G1S cyclin and CDK will pair with each other and they become active, and they will go and they will phosphorylate some target protein. Remember that cyclin dependent kinase will be active upon binding to cyclin, and then the combination of cyclin CDK will go and activate some other protein by phosphorylating them. So remember, this general scheme will be cyclin and CDK. Now let's say CDK is inactive. Now once they pair with each other, it is active. It becomes active. Now this active cyclin CDK complex can go and phosphorylate any other protein, and that phosphorylated protein will phosphorylate some other stuff, and then the cellular progression will take place to the next level. Okay, and in this case phosphorylation required in many different aspects, many different accessory proteins there, different enzymes are there which are activating for, say, the DNA replication and proper maintenance of the size, the different actin filaments and stuff. So all these things, this phosphorylation is very, very important stage which will carry the cell to the next level of cell division, and it's required, and this process is kind of same for all the cyclins that are present here. Now one thing I must tell you, the thing is, these cyclins are specific for each phase, right? For example, G1S cyclin will not function at G2S phase, G2M phase, or S phase cyclin will not function at M phase. They are specific for a specific phase of cell cycle. Why so? Because let's guess the cell will go and divide again and again. So if G1S cyclin remains here at the S phase, then they will again turn the G1S phase on. Then again try to put it into the S phase. It's not required, right? If it presents there, if the cyclins are always present and active, because if cyclins are there, CDKs will be active. So if cyclins are present and make CDK active, then they are going to activate whole lot of proteins all the time inside the cell, and then what will happen? Some of them will try to drag it from G1 to S, some of them will try to drag it from G2 to M, some of them will try to drag it from metaphase to anaphase. So it will create a war inside the cell, right? Or it will cause the cell to divide multiple times even without any kind of checkpoint hints, and the cell definitely not want that. So what the cell will do here is that's why produce specific type of cyclin partners for CDKs which will work in a specific time only. So G1S cyclins only work at the G1S phase, G1 phase. Cyclin CDK, which is SG2 or S phase cyclin, only work at S phase. G2M phase cyclin only work at the end of G2 phase, and then the this cyclin B and CDK complex, or we also known as mitotic cyclins or metaphase cyclin, it will only work at that stage. Okay, so these are the stages where those cyclin partners are present, then only it will activate CDK, then CDK will take action, then the sequential events will happen and the cell will progress to the next level of cell cycle. So this is the basic overview. So what we know, at each time and at each point, let's say G1S cyclins are there. Now they are activating CDK. Now the CDK is now active. CDK will go and phosphorylate some proteins to drag the cell cycle from G1 to S phase. Now once it drag it from G1 to S phase, after the task is done, then when it is in the S phase or synthesis phase, then it should be taken up by the S phase cyclins. So all G1 S phase cyclin, all the G1S cyclins, the previous type of cyclins or the cyclin Ds will be degraded. So the earlier cyclins are degraded when it enters to a new phase of the cell cycle. Similarly here, when this S phase is over, it will bring it to the next phase. All the S phase cyclins are degraded and they will start making the newer cyclin, that is cyclin B at the end. Okay, so that is the idea. So if you check, but the CDKs, if you look, CDK is present throughout the cell cycle in the same concentration. So if you check for a cell, what will you find is that you will find a steady rate of CDKs throughout the time, while the cyclins will rise sometimes, fall sometimes, rise sometimes, fall sometimes. Some specific type of cyclins rise at a point, let's say G1 phase, this is S phase. So specific cyclins rise at a point, then again they are degraded. Then again another type of cyclins find and they are degraded. So if I draw it for different cyclins, what it will look like? It will look something like this. Steady rate for CDKs, but for the cyclins, let's say G1 cyclins, we have G1 phase, S phase, and let's say M phase for first. So G1 phase cyclins, if you see, G1 phase cyclins will rise and then this. S phase cyclins will be very less and it will rise. M phase cyclins will be rising at the end. So you'll see different type of graphs for different type of cyclins, but the CDK is always present at the nominal level. This one, this is for the CDK, and these are the cyclins. Okay, and that is why it's important, because the old cyclin should be destroyed, because if the cyclin is present, then obviously CDK will function and they don't want that. Now let's come to the one example of how cyclin CDK partner works and how they activate or deactivate stuff. Okay, so let's say here the activation process of cyclin CDK complex is not very easy. For example, here they have a kind of autophosphorylation and dephosphorylation events going on to make them active or not active. For example, if you take M phase cyclin and CDK complex, that is known as a maturation promoting factor or mitotic promoting factor or MPF. It is known as MPF because they are promoting the mitotic stage. So here we have this cyclin, and we have this CDK. Okay, now the idea of cyclin CDK is slightly different here. They require different combinations of proteins to work together to give this result. Okay, so this maturation promoting complex formation is itself a difficult task. So cyclin, and this is known as the M phase cyclin, remember, or cyclin B. Okay, and this is the CDK of M phase. Actually you can't say about phases in CDK because CDK is present throughout the time, but this is M phase cyclin. Now the cyclin will come and attach to the CDK. Now CDK is not functioning because it will not function without cyclin, and cyclin cannot do any task on its own. So cyclin will come and pair with CDK. This is the first stage. Pairing takes place. After the pairing is done, then there are kinase molecules. Okay, two different kinase molecules will involve and they will try phosphorylating CDK. Remember, CDK itself is a kinase, but normally when it is bound with cyclin, it is not active. They need to have some sorts of autophosphorylation or any sorts of phosphorylation from other molecules to become active. So this CDK is inactive, though it's bound with cyclin now. Now it requires other phosphorylating enzymes, kinase enzymes, and there are two different kinase enzymes. One is called V1, this is called an inhibitory kinase. Another one is called CAC. This is activatory kinase. Inhibitory kinase and activatory kinase. Both of them kinase proteins. Both of them have the capability of phosphorylating this CDK. So V1 phosphorylates is this location. We call it as a phosphorylation. Let's write P. CAC also phosphorylates in another part of that CDK protein. So if you look at CDK protein, it is there are different amino acid sequences. There are specific amino acids denoted where V1 phosphorylate as well as CAC phosphorylates, but they phosphorylates in different locations, different amino acids in the cyclin-dependent kinase in the M phase. Now still that point, CDK is dual phosphorylated, bound with cyclin, but it's still not active. You know why? Because it's phosphorylated with V1, which is the inhibitory phosphorylation. That means if this V1 phosphorylates this CDK, and if this phosphorylation is present, that CDK will not become active. The CDK will only become active if if V1 phosphorylation is taken away from it. So if we dephosphorylate the V1 phosphate. Okay, so after that what will happen, the ultimate stage to make it active, when everything is fine, they check everything is okay, the metaphase checkpoint is fine, the metaphase arrangement of the chromosome is okay, microtubules are properly connected to kinetochore, and all these things are fine, then they will add this final signal of a protein called CDC25. CDC25 is a phosphatase enzyme which will cleave phosphate group from protein said. So here it will cleave the phosphate group of this V1 phosphate. So now they carry only one phosphate at CAC end. So now it becomes activated. Now this cyclin CDK complex will be known as MPF, maturation promoting factor, which will bring and activate other proteins like cohesin and stuff, and they will help in the segregation. Condensin and cohesin and all these other proteins will help in the separation of those chromatids from each other. So that is the series of events. Remember, now you may ask me a question that why is the requirement of dual phosphorylation in the first place? If you need to activate cyclin CDK, you can simply direct use CAC and phosphorylate that. It will become active. Now the answer is, in biological processes, it's never granted that you get something. It's always, the door is always closed. It is only open if you show your identity card. That is the idea from a biological perspective. It's not like that if the door is open, now once you're going, if something is wrong, you will close the door. It's not true actually, it's not fine, it's not the good way to control something. If you want to control the uptake of something, you should close the door all the time and only open it when the person has the ID card. Just put the analogy here. Here it's already blocked, everything is prepared at this end, because we require a lot of this CDK cyclin complexes here at this M phase to work properly, because we need a lot of phosphorylations in the next rounds for the cell to divide properly. So what we'll do here, we produce a lot of cyclin CDK complexes and we do this dual phosphorylation to all of them. So all this huge concentration of cyclin CDK complex is present even before the checkpoint is assured whether everything is fine or not. So there are two tasks going on. One is to checking whether everything is going on or not. We are finding things, whether the microtubule assembly is fine, whether the alignment of the chromosomes in the metaphase plate is fine. It's checking things. Meanwhile, it's producing cyclin CDK complex for MPF. It's producing it, but it should not make it active. It blocks its activity, it holds it to this double phosphorylated form. It places it here. So 3 of the stages are already converted and the proteins are present in this format. So now they are standing there for only one order. The order is everything is okay. If the order is okay, one dephosphorylation will take place and all of a sudden all those CDK become active and the maturation promoting factor will work, cell division off fast. So if they stand there at this point to get the idea whether everything is fine, then they'll prepare this thing, it will take a long time. right? Because the cell division takes place very, very fast. You know how fast it takes place, right? It's very fast, incredibly fast, and lot of proteins to be made. So the only way to do that, you prepare it and put it in the ready-made fashion, but you use it exactly. So if you check that MPF concentration, if you check a graph of MPF concentration, it will give you the idea. It will give you the basic idea here, because in the graph you can see that at the end, at the time of M phase, at the start of M phase even, and at this G2 phase, at the start of M phase even, you will see the concentration is very low. But this is the midpoint of M, this is the anaphase. Just try to think of this. Even the start of M, the concentration is very low. But right after the situation, the signal is okay, signal is positive, you will see a very sharp peak. A very, very sharp peak of this cyclin CDK complex. This is MPF. So if you look at this graph, how can you say, what can you say is that the concentration increases very sharply, very rapidly when they get the positive signal. And that is possible due to this dual phosphorylation event. Okay, so that in a sense is how cyclins and CDK work together to make other proteins activated and to pass a cell from G1 phase to S, then S to G2, G2 to M, and then finally the cell division is done. So these are the sequential events, and I hope this video helps you. If you like this video, please hit the like button, subscribe to my channel. This is the button here in the bottom, in the top, everywhere. Just hit the button to subscribe to get more video updates like that. Thank you.